In this video, we're going to talk about some of the ways that eukaryotes can regulate transcription by modifying the structure of chromatin. So what is chromatin? Remember back to earlier in the semester. Chromatin is a structure that consists of both DNA and protein components that functions to condense the DNA so that it can fit within the nucleus of the cell. A key part of chromatin structure is the presence of nucleosomes. These nucleosomes are formed when DNA wraps around a core of histone proteins. So rather than the DNA being a long linear molecule, it is wound up and coiled. The nucleosomes can then be packed together to further condense the chromatin. So what exactly does this mean when it comes to transcription? Well, it may mean that depending on the degree of DNA condensation, the regulatory and transcriptional machinery may not be able to bind to the DNA. So how exactly does the cell gain access to these sequences? Well, to explain this process, let us look at an activator. This particular activator is looking to bind at the regulatory promoter of a gene in order to increase gene expression. But both the promoter and the gene are located within a region of DNA that is highly condensed, meaning that the nucleosomes are packed together tightly. Because the DNA is wound up so tightly, the activator cannot bind, which means it cannot work to turn on gene expression. So what can the activator do? The activator can recruit enzymes that add acetyl groups and remove methyl groups from the tails of the histones in the nucleosomes. By modifying the histone proteins, the position of the nucleosomes can be changed, allowing for the DNA to become less condensed. This will allow the activator to access the necessary DNA sequence. Let us look at some modifications that can be done to histone proteins in order to change the positioning of the nucleosomes. One modification that can be done is acetylation. An enzyme called histone acetyltransferase can add acetyl groups to histones. The addition of acetyl groups removes the positive charge of the histones and causes them to be less attracted to the negatively charged DNA. As a result, adding acetyl groups causes the DNA to be less condensed. This is one way that results in the chromatin structure becoming more relaxed, allowing the activator to bind to the DNA. Another way that the chromatin structure can be relaxed is through demethylation. Removing methyl groups from the histone proteins allows the nucleosomes to reposition further apart from one another, allowing for greater access to the DNA. Great, we have solved the problem. The activator can now bind to the DNA and transcription can move forward. But wait, not so fast. There's another problem. What happens if the gene's core promoter is wrapped around the nucleosome? This means that the correct DNA sequence is all wound up and cannot be accessed. How does the cell get access to these regulatory sequences? Well, the activator can solve this problem too. Just as the activator recruited enzymes to help modify the histones, the activator can also recruit a protein complex that can reposition the nucleosomes. This protein complex is called SWI/SNF. After being recruited by the activator, the SWI/SNF complex can shift the nucleosomes, exposing the regulatory sequences so that they are no longer wound up on a nucleosome, allowing for regulatory proteins and transcriptional machinery to bind. After these modifications, the activator, along with all the necessary transcriptional machinery, are finally able to bind to the DNA and turn on gene expression. So now you know 3 different ways that eukaryotes can modify the chromatin structure in order to regulate transcription. Thanks for watching!